Hello, I'm Dr. Christo Zuves, the Medical Director at Zuves Fertility Center in Foster City, California. Here at ZFC, we are always looking for ways to pick the best embryo for transfer to maximize success. We were excited when it was suggested that mitochondrial DNA levels could predict an embryo's clinical outcome. This paper summarizes our findings on this subject.
Hi, I'm Andrea Victor. I'm an embryologist here at Zuves Fertility Center and also part of our genetics team. A couple of recent high-profile papers have reported that the mitochondrial DNA levels can predict the implantation potential of an embryo during IVF. The suggestion has also been made that aneuploid embryos have higher amounts of mitochondrial DNA and that embryos derived from eggs retrieved from advanced age patients also show higher mitochondrial DNA levels. In general, these findings correlate a stressed embryonic state with high levels of mitochondrial DNA. This agrees with the quiet embryo hypothesis, which postulates that embryos attempt to compensate for stresses by elevating their metabolic activity. Our study can be divided into two parts. In the first, we delineate an accurate way to determine the levels of mitochondrial DNA in blastocyst biopsies. In the second part, we apply those improved calculation methods to data from our laboratory. Mitochondrial DNA levels in any biological context are generally determined by next-generation sequencing or quantitative real-time PCR. In its simplest form, one can count raw mitochondrial DNA molecules and compare those numbers between blastocysts. But this comparison is distorted if the biopsies contain different number of cells or there are differences in efficiency of downstream technical processes. In order to circumvent that, studies have used a count for nuclear DNA as well, taken ratios of mitochondrial DNA to nuclear DNA, and then compared these ratios between embryos. The issue is that when dealing with aneuploid embryos or embryos of different genders, the amount of nuclear DNA is not equal between blastocysts. We have developed a mathematical factor that corrects for these differences, such that we can do true normalized apple-to-apple comparisons between embryos. This slide explains the rationale for the correction factor when using next-gen sequencing to determine mitochondrial DNA levels. Assume we have biopsies from 3 embryos that all have equal levels of mitochondrial DNA. For argument's sake, each cell yields 1,000 reads aligning to mitochondrial DNA and 100 reads aligning to nuclear DNA. For embryos A and B, we obtain the same ratio even though embryo A contains 6 initial cells and embryo B contains 4 initial cells. But for embryo C, we obtain an artificially inflated value because the initial amount of nuclear DNA is smaller, considering this embryo is missing chromosomes 1 and 22. Also, being male, instead of a second X chromosome, it has a Y chromosome, which is shorter. We must adjust this skewed result using a correction factor. The factor is tailored to the composition of the nuclear DNA of said embryo. The more DNA it is missing, the larger the correction factor. In the manuscript, we provide a full list of correction factors for all chromosomes that might be monosomic or trisomic. We also outline a universal formula that can be applied for any possible full chromosomal or segmental loss or gain to any monosomy or degree of polysomy. We also developed a correction factor when using qPCR. Assume the reference assay for nuclear DNA targets a region that is disomic. In that case, the correction factor is 1. But in an aneuploid embryo, that region can, for example, be monosomic or trisomic, and we need to adjust for that by the indicated correction factors. If the nuclear DNA reference assay targets a region that is nullisomic, we must choose a different reference assay altogether. Extending this logic, when comparing euploid embryos, if the nuclear DNA reference assay maps to a region of the X chromosome, we must apply a correction factor when comparing male and female embryos. In the second part of the study, we investigate levels of mitochondrial DNA in blastocysts that were used for IVF in our clinic. We analyzed embryos by next-gen sequencing as well as by qPCR, testing 2 different mitochondrial genes and using 2 different analysis methods, absolute and relative quantitation. Surprisingly, when we group blastocysts by euploids and aneuploids, we don't see a statistically significant difference in terms of mitochondrial DNA levels. We detect a range of values, but the range is similar across both groups. When we break blastocysts down by oocyte age, we also see no significant trend. Of major clinical relevance, when we compare euploid embryos that successfully implanted and those that failed to implant, we again observe no statistically significant differences in mitochondrial DNA levels, which is in stark contrast with previous reports. In conclusion, we recommend that labs analyzing mitochondrial DNA levels in embryos make use of the accurate determination methods we laid out in this paper. Our in-house embryo data suggests that mitochondrial DNA levels do not correlate with blastocyst ploidy, age, or implantation potential. We hope that future work from other independent laboratories will help clarify the relevance of mitochondrial DNA levels as a biomarker for embryo viability. Thanks for watching.